science teacher. I'm going to be going over how and why you all should be doing botanical research for your projects. Try to sell you on that, get my seminar in early in the year, early in the summer, so you all can start doing plant projects and ignore all that biomedical stuff that we learned about yesterday. But that's my goal at least. All right, so I feel like I should go my background a little bit. So before I was a teacher, I wasn't always a teacher. I didn't go to school to be a teacher. I did my degree in spelling error. I did my degree in biology. That's starting off right. Biology at Penn State. And before I started being a teacher, I worked as a field botanist. I worked at the Smithsonian Museum of Natural History in DC. Did you guys ever been there? I saw all the... When you guys go to the museum, you see a lot of specimens, like exhibits on display. That's only a quarter of what a museum offers. I was kind of behind the scenes, working in the research collection. So I worked as a researcher there in the National Herbarium, looking at molecular biology work there. And then I also worked as a botanist and lab tech in Florida for a little bit. And then I got an education back after that. And here's some me with, I like posing with really big plants. I had another one I couldn't find, but this was a really cool, this is actually the plant that they ferment tequila from. This is an agave. I didn't take a picture of it because they ferment it into tequila, but because this plant only flowers about every 100 years. So it takes 100 years before it flowers. It sends up this giant flower spike, spends all its energy in making that flower spike, and then the whole plant dies. It's gone. Now, why would a plant spend all its energy in making this flower spike to a point in which it dies?
To make more plants.
To make more plants? All right. So yeah, this is a really big energy expenditure, right? So it's putting all its effort into making offspring, passing its genes on to the next generation. And for that plant, for that specific species, it's successful enough that it's able to produce more offspring to go into the next generation. So it doesn't mind that it dies. It works. It's successful. And this was me. I was doing ecological research in Costa Rica. This was me in a cloud forest. It was a very weird experience in a cloud. It's cold, but it's also really humid. A really cool plant life on here. You can't really see with the lighting. So what I'm going to be talking today, there's kind of three parts. First, I'm going to be going over why I think plants are fascinating to study, why you should all be studying them with your independent projects. Then I kind of threw together some ideas that I think are cool. I'm going to go over some general subfields in botany and some project ideas that I would love to help you with, or you can take from me and run with it. And then I'm going to give you some places to look for more information and ideas. All right? So first, why study plants? First, their diversity. So this is a plate, a really cool plate from one of my textbooks that I have that I always go back to. It just kind of illustrates the types of plants that we have in existence today, kind of like the living diversity. So you have something as simple as a algae species up here, to something as complex as an orchid that mimics female pollinators that kind of get pollinated. You have conifers. This guy here, this guy's name is Welwitschia. He only grows in Peru, in South America. And to me or you, that may look like a wilted, kind of dead, grassy kind of plant. But this guy is actually closely related to kind of like a pine tree, kind of like something you would set up for Christmas. So this is really cool plant guy here. A lot of interesting diversity. This guy's a ginkgo. Really cool leaves. Ease of growing, so they're pretty easy to keep. They're not like culturing another organism that you have to kind of revolve your life around taking care of. You can grow these in the greenhouse, you can grow these under lights, and for the most part, they're pretty easy to take care of. There's less paperwork involved with them. So if you do any research with kind of animals or insects or bacteria, there's always extra layers of paperwork that you have to fill in order to present your work to scientific conferences, science fairs, kind of like safety paperwork. That doesn't really exist with most plant species, so it's less work on you in terms of kind of like sharing your research with others. And then the broader impacts. Plants are really important, obviously. It's really easy to tie your research into making it kind of like the who cares factor of your research. So while you may be interested in, say, this rose and the factors that may affect how big it grows, you need to be able to sell that idea to people other than yourself, making a pitch, making other people care. Why should anyone care about your passion project? So I think that the broader impacts of plant research is there. So the first area that I often see with mentoring students and working in this area are going to be your plant growth and development research projects, where you're looking at what is the effect of X on plant growth. Common research project, and plants work for these because they do something called indeterminate growth. That just means that a plant is growing a new plant throughout its entire life. So if I were to take a cutting of this plant, chop off that stem here, stick it in the dirt, the cells at the tip of that plant would grow and keep on making a new plant. If I took Xander and cut off his arm and put it in the dirt, would I grow a new Xander? No, it doesn't work. So plants are really cool in that they're able to keep on growing more and more plant throughout their lives. Okay? As humans, you guys are all growing, but at a certain point in your life, you're done growing. Okay? We can't generate new limbs, new parts of your body for the most part. There are some organs that can regenerate, and there's research being into that area. But plants can grow more and more growths. And it's easy to manipulate a factor in the plant's environment, and then you can quickly see how that affects how quickly the plant's growing. But the one kind of thing you have to be careful of, this is a common area of experimentation. If you're interested in designing a plant growth experiment, you need to make sure that your project is novel. What does that word novel mean? If I say I have a novel idea. So novel is going to be new or unique, right? If I was going to look at the effect of fertilizer on plant growth, do we know what's going to happen there? What's going to happen when I add fertilizer? It's going to grow quicker, right? Not a real novel idea per se, but you can change that idea to make it more unique and stand out. So you have to be careful when you're doing these kind of experiments. A lot of them haven't been done before or done a lot. So you need to kind of add the next layer onto that. So some research questions that you could design related to plant growth. What is the effect of blank environmental factor on plant growth? Obvious. When you're designing these kind of research questions, you need to focus on the broader impacts on the environmental factors. So if I go back to the fertilizer example, who cares about the effect of fertilizer on plant growth? Who would care about that? Who would care about you doing an experimental work where you're looking at the effect of fertilizer on plant growth? Yes?
Farmers.
Farmers, right? So you need to sell your research idea in that you're interested in looking at fertilizing your plants and seeing how they grow. You needed to relate that to people outside of yourself. So applying it to agriculture, production of agriculture, getting crops. So when you are conducting your plant growth experiment, you want to avoid simply just measuring the height of the plant. You want to simply just avoid just measuring centimeters. Okay? That's an easy one to do. You need to go an extra step beyond that. So some examples that you could look at when you're quantifying plant growth. Cell wall size, leaf diameter, chloroplast, number of chloroplasts in the cell, root growth, flowers, all examples of things you can measure to see how your environmental factor affects the growth of the plant. You want to simply avoid, just measuring plant height. Okay? You want to add that next layer onto this. This is a common experiment that I see. What's going on in this experiment? Anybody see that picture? What are they pouring on the one plant and not on the other? Yeah. What is that? Okay, that one's getting water, right? What's getting poured on that one? Yeah? Soda. Okay? What is the effect of soda on plant growth? Anybody ever see that experiment? Maybe you've done that experiment. Okay, maybe you've seen it at science fairs, all right? Don't do that. Okay? So when you're looking at the effect of soda on plant growth, who cares about that experiment? Are people concerned about watering their plants with soda? Is that a major area of concern in the environment? Is it raining soda from the sky? No. So how could you change this experiment where you're adding soda to this plant? How could you change that to be more scientifically accurate or more impactful? Yeah.
I guess if you like poured fresh water or like salt water on plants.
So you could change this soda aspect. So when you're adding soda, you're really looking at the effect of maybe the added salts in the soda, the acid in the soda. So you really want to go and focus on that aspect of this and not simply just pour soda. All right. Next Wednesday, I'm going to be going over how to set up and kind of carry out a plant growth experiment like this. We're actually going to be setting up our own. You're going to be designing an experiment, collecting data. I'll be going over how to do some of these kind of data collection steps. You guys are going to be designing and growing your own. I did have to change it because of timing, so it's going to be next Wednesday, 9 to 11. It is limited number of seats because we are doing a lab in the classroom, so if you're interested, sign up. Next, plant physiology. This is going to be looking at how a plant functions. So plants are stationary. I put an asterisk there because they really are moving, but they're kind of stuck in place, right, with roots. So the plant has to accomplish all of its life functions in one place. When we're looking at plant physiology or how it works, the scale of this kind of work is going to range. So it's going to be small. You could be looking at the molecular interactions of water and photosynthesis, right? I know my guys, and if you've taken my class, we've talked about photosynthesis and the reactants and the products. And if you've taken biology, you're tired of hearing these. But there's a lot of interactions between the cells of the plants and how they're able to take up water, how that water is transported around the plant. You could focus on the small scale of plant physiology, or you can zoom out to a larger scale. So how hormones control plant development. You know plants have hormones. Plants are giving off hormones at all times, and these hormones control plant development at different parts of its life cycle. We can go and buy bottled plant hormones and expose it to our plants. So we can look at hormonal control of plant growth and development. How plants respond to the seasons. They go dormant, how they reproduce. Francesca, what's up?
So now we're doing about like how a plant functions and grows. Since there are like mutations in human DNA, could there possibly like be mutations in plant DNA
All the time.
that affects the way?
All the time. Yeah, so plants are being bombarded by light every day, right? As the light strikes the cell, just as how light strikes our cells, it can mutate our DNA. Plants are dealing with that all the time as well. Yeah. So what happens? Some research questions. So these are ones that I came up with. I thought would be cool to look at. So how does the chemistry of the soil affect the anthocyanin pigmentation in the pansy? So here's a pansy flower. These are the ones that kind of grow early spring. So how does the soil chemistry affect the coloration in the petals? Right? Who would care about that kind of project? Xander. Oh, not Xander.
Forests.
Florists, right? People who grow plants for their ornamental value. How does the addition of plant beneficial microbes influence infection by plant downy mildew? This is a plant downy mildew. It's that white kind of powder on the leaves. If you look outside right now, because we've had so much rain, a lot of plants have this kind of white growth on the leaves. That's a fungal infection. So you can go to the store now, kind of like a big fad right now is adding kind of a plant probiotic to the soil, just how you and I can take a probiotic in yogurt with Activia yogurt, or go buy a probiotic pill. You can get probiotic supplements for the soil, give it to these plants. The idea is it makes them healthier. So how does a plant probiotic affect the infection by this plant, downy mildew? Cool experiment, plant pathology. And then how does competition between plants impact the rate of water loss? Some ideas. This is where I did my work in undergrad. I did my undergraduate research in plant and insect ecology. I These are some caterpillars going to town. So plant growth is not a solo act. Plants are interacting with insects constantly. The two major categories here are going to be pollination and herbivory. What is herbivory? Hey, Jessica?
The insects eating the plant.
Yeah, whenever something eats a plant, right? Plants serve as both a food source and a potential site for egg laying. You see insects eating plants all the time. They also lay their eggs on their plants, like this right here. So when these guys hatch, they have a food source that they can go to town and munch on. Food preference. You can look at the food preference of different insects in the lab by doing something called a choice test. This in the diagram here is what's called a choice test, where you place a bunch of diet options around the dish. You place your insect in the middle, and then you watch, and you see which one does the insect prefer. And it's really cool when you place a caterpillar kind of in the middle here, you'll actually see him kind of rear up, and he's kind of sniffing the air almost, sniffing out for his diet and seeing where his tasty snack's going to be. And then you observe what's his preference in the dish. Does he prefer to eat one plant over the other? Series of trials and observations you can do here looking at the food preference. And then you can also look at egg laying preference. Right? Moths and insects are going to prefer to lay their eggs on certain plants over others. For instance, the monarch butterfly. What plant does the monarch butterfly prefer to lay its eggs on? You know?
Milkweed?
Milkweed, very good. So how does the monarch butterfly know what a milkweed is? Well, it kind of can sense the presence of the milkweed, looks for volatile cues in the air, and it smells it out, it finds it, and lays its eggs on it. So you can do these in observations in the lab or in the field. Insects and pollination. I'm not really going to talk about this right now. Mr. Searsboss has a session on the 22nd, I know, where he's going to be looking at plant pollinators. So if you're interested at all in pollinator biology, come check out that session. Really cool lab work you can do when you're looking at how insect pollinators interact with plants and why they're important. So some ideas you can do with this. How does plant activity, so native versus non-native, impact the herbivory by the invasive Japanese beetle? You guys see the Japanese beetles are starting to emerge, yeah? Those shiny beetles that kind of eat up all your leaves on the plants. They kind of make the leaves look like skeletons. Well, do those beetles have a preference in food source? That would be an interesting question to ask. How does prior herbivore damage, so does being munched on by an insect impact where the spotted lanternfly lays its egg? Does the spotted lanternfly prefer to lay its eggs on a plant that's already been chewed on versus one that hasn't been chewed on? What benefit would the spotted lanternfly have by laying its eggs on a plant that hasn't been chewed on? Xander? Kids would have plenty of food, right? That'd be a cool question to ask. Be cool. I don't know. And then finally, how does competition for feeding sites impact plant food preference of the tobacco hornworm? Tobacco hornworm here, big green gnarly guy. So it'd be interesting to see, will this guy prefer to eat a plant that's already being munched on by his brethren, or would he prefer a fresh one? Cool question to ask. Molecular biology. So we can easily access plant DNA, and I know Mr. Seifert has been talking about the DNA barcoding. You might have heard him talk about this before. But we can actually identify plants through their DNA sequences. We call it DNA barcoding. We can extract the DNA from a species, sequence it, and then identify that organism to the species level. Really cool questions you can ask there with DNA barcoding. You can also screen plant compounds for medical uses, antioxidants. Plants are producing a ton of these that we can take advantage of for our day-to-day lives. And then DNA transformation. DNA transformation is just inserting a foreign piece of DNA into a plant. So here we have a firefly. It glows because it makes the protein luciferase. We can take that gene for luciferase and stick it into a plant and make the plant glow. All right. Some research questions. I think this one's really cool. We can DNA barcode. It'd be cool to DNA barcode the picture of a carnivorous plant to determine what it eats. So we have some of these growing out in the garden. We can take out its guts, sequence what's in its guts, and see what insects, what food sources is this pitcher plant eating. Is it only catching certain things? Is it catching a wide variety? What stuff's in the garden? What effect does canning have on the antioxidants of grapes? And then DNA sequencing of ornamental plants would be interesting to look at. And then finally, diversity and taxonomy. This one's kind of often ignored. So developing the ability to identify and classify organisms in the field, that's an invaluable skill set. By going out into the field and being confident enough to identify things down to at least family level. That's in-demand skill that's often not taught anymore. These broadly trained taxonomists, so they're in high demand by many different organizations. So being able to take a book, take yourself out in the field and identify plants or identify any organism is always a useful skill. These are often developed in this kind of botanical research. So it's often developed in any kind of research project. You're learning how to identify these organisms in the field. You could take and develop a diversity and taxonomy study by surveying levels of species richness and abundance in the field. Some questions that you could ask in these kind of in this area is what effect does field pesticide spraying? What is the effect of field pesticide spraying on the diversity of the forest edge? So we're spraying a ton of pesticides on our athletic fields to keep the weeds and insects down. But what effect does that pesticide spraying have on the diversity of the creek and forest life out back? It would take someone to go out there and actually identify and quantify the plants that are growing near the school and seeing what that answer was. And then how do various logging practices affect the species richness of understored plants? Just sort of questions you can look at in this field. So if you're interested in doing research in this area, some sources you can check out. There's science societies you can look at. So the Botanical Society of America, the American Society of Plant Biologists, both have really good web pages with lots of information. Some stuff that's really digestible chunks that you could look at. Those are both available to you. You just Google American Society of Plant Biologists or Botanical Society of America and they'll pop up. Some good academic journals. You should have access to all of these that you can look through. Some of these may be a bit higher level, but I know Botany and Plant Direct are a little bit more digestible. Good chunks of information that you could skim through, abstracts and seeing what catches your interest. So a lot of areas that you can look at, a lot of sources you can check out if any of these kind of topics interest you. Now, I'm done for my talk. Are there any questions looking at these areas? Anyone consider doing a plant biology experiment for their research project? I know Francesca's in it, she's in it. All right, very cool. So if you're interested still in conducting a plant biology experiment, guys, next week, my seminar looking at, we're going to be designing these experiments and carrying them out in the greenhouse and grow room. If you're at all interested, sign up for that. We're going to be doing some lab work with that as well, growing the plants and then taking measurements over the rest of the program. Okay?